but a big welcome, everyone. Thank you very much for making time to join today's webinar on optimizing oncology diagnostic trials. So my name is Mary Beckwith, co-founder and CEO here at Lindus Health, and I will be your host for the session. Just to set the scene, and then we'll hand over to our panelists. So liquid biopsy and oncology diagnostic trials generally, bring unique challenges. This is a very distinct kind of trial operationally. Different designs, different populations, certainly different regulatory challenges compared to, say, drug trials. And so today we'll hear from our expert panel. They'll share some of their in-depth experience of navigating these challenges. Yeah, it should hopefully be a great session. Quick bit of housekeeping before we kick off. So we will have a Q&A at the end, but as we go, please do feel free to drop your questions. There should be a Q&A box in the bottom of the screen. It's not appearing for me, but I think you guys have a different view than I do. So yeah, please do type in your questions as they come up. I can try and MC and throw them into the discussion if they're relevant. Otherwise, we've got time at the end to make sure we get through as many as possible. So with that, thank you all very much. And I will hand over to our panelists for introductions. Eric, do you want to go first?
Sure. Mary, can you hear me?
Yes.
Great. Hi, everyone. And Mary, thank you for allowing me to attend this. So my name is Eric Haltom. I am the Senior Vice President of Clinical Operations at Harbinger Health. Harbinger Health is actually located in Cambridge, Massachusetts, so I'm in the office today here in Cambridge. I have been in clinical operations for 30 plus years. At the beginning of my career, I started in academia, coordinating clinical trials for the genitourinary division at the Dana-Farber Cancer Institute. Since that time, I've been involved in both small and mid-sized biotech companies. I've worked extensively on oncology and rare diseases in most of my career. And although Mary might have outlined Hutan and I as an expert, I can't say that I'm an expert in the liquid biopsy space, but I think I give a good sort of dynamic on comparing therapeutic trials and interventional trials to the liquid biopsy space. I have been at Harbinger Health, if I hadn't said that, for a couple of years now. I've had the opportunity to build clinical operations, data management, clinical outsourcing teams, and clinical supply teams for the past 15 years or so in my career. So it's a pleasure being here with you all, and thank you, Mary. Hutan, I'll hand it over to you.
Thanks, Eric, and thank you, Mary. It's always a pleasure to speak with colleagues who are like-minded. My name is Hutan Ashrafian. I'm a clinician scientist. As you can tell from my accent, I'm a Brit, and my background is an esophageal gastric surgeon who's also trained in cardiothoracic surgery. And I've been in clinical practice for over 20 years and have always been pushing in the ways that I best could innovations that I could bring directly to patient care. So I initially became a full-time surgeon, then I built a lab that was focusing on introducing diagnostics into patient care. And I worked with some of the big tech companies to try and get AI solutions into diagnostics, particularly in cancer patients. I was focusing on cancer in the sense that it was something that was not only a source of massive pathology in the world, so that, there's approximately 20 million cases a year, but the issue, and maybe 10 million deaths a year from cancer. But the reality is that the global prevalence of cancer is almost doubling. And so by 2050, we'll be maybe doubling those numbers in terms of up to 40 million and 20 million deaths per year. And there was something that was discordant in that space. So I decided to try and find how I could optimize and get really fast in finding solutions. And so I became a doctor who also worked in industry. And it's through that I entered the world of flagship pioneering, which is this big venture incubator where lots of companies are created. And through there, I was introduced to Harbinger Health, which is where I am now as their Chief Medical Officer.
Nice. Awesome. And I'll give a quick introduction. So Mary Beckwith, co-founder, CEO at Lindus Health. We help companies run radically faster, more efficient clinical studies. We have kind of diagnostics generally, and within that, oncology diagnostics is a big area of focus for us. We're lucky to have a number of clients, of which Harbinger is one. And so our perspective, mostly comes from the kind of nitty-gritty operational side. But very grateful to have Hutan and Eric here as our experts to take us through this. So perfect. So as usual, we kind of kick these off with a bit of a poll. So I'll go ahead and launch that now. Cool. So you should all have a poll up on your screen. We're just kind of curious to find out kind of where you as our audience sit in terms of your kind of development journey. It helps us calibrate the rest of the talk. So yeah, go ahead and fill that out, and then we'll have a look at the answers in a moment. Cool. Give everyone a couple more seconds. Okay, perfect. Let me close that and share results. So yeah, it looks like obviously kind of as expected, a big mix of folks with majority of people in the kind of earlier stages of either planning a validation trial or running a study. So yeah, that's a pretty good match for what we expected and some of the topics we'll cover today. Cool. Great. So yeah, I think firstly to kind of set the scene, when we think about oncology diagnostics and really cancer care, diagnostics can mean different things depending on where a patient is in the care continuum, and obviously where your diagnostic sits in the care pathway can have pretty big downstream consequences on your validation journey, intended use, and so on. So yeah, I mean, Hootan and Eric, could you give us an overview of how the field is tackling these challenges? How diagnostics need to differ depending where in the care pathway they sit.
Yeah, I mean, maybe I'll start and then let Eric come in. And so the cancer care continuum, of course, encompasses quite heterogeneous groups of individuals. And for example, our proprietary test is designed to Harbinger is designed to maybe look at many of these steps, but each one has its own specific needs. And so the nature of addressing a diagnostic paradigm in each of these spaces brings with it its own unique sources of success, but also areas that need to be addressed. So, for example, in a epidemiological type of screening setting, when one is looking at diagnostic accuracy, the nature of the diagnostic accuracy, let's say study type would be distinct from that of an MRD one, where, for example, in an MRD setting, statistically, you'd power yourself considering prognostication outcomes, an improvement of longevity, maybe with concepts such as Cox regression and and randomization to what is superior or non-inferior. Whereas let's say if you're doing a screening study, and we've been involved in kind of setting up one, we look more for, let's say, a prospective cohort of individuals and how to follow them up, or maybe think about doing a pragmatic cluster RCT, whereas in a classical diagnostic cancer test, it would be that classical RCT diagnostic accuracy and strategy paradigm. So each one of these things brings its own particular needs, and so anyone entering this space would need to kind of not only think about the study size of what their primary endpoints are, but actually what the underlying incidence of the condition is. And as you can see, for example, early disease might go up to a prevalence of maybe, or aiding diagnosis might be a prevalence of maybe 10%. In asymptomatic individuals, it might be something like 1%, 1 to 2%. And then in those who you're monitoring cancer, you already know that there's a cancer in those individuals and you're looking for metastases and spread. And so you've already had an inference based on where they started biologically. And so for each one of these things, you need to kind of bespoke your approach to your clinical question and your evidence generation.
Yeah, makes sense.
Yeah, I'll just add, and most of the responses I have today, Mary and team, will be really through the operational lens and executing on that. And so when I look at this continuum, these are all differing populations. And from an operations perspective, what types of sites are you targeting for each of these populations? So when I look at the screening for cancer, are you looking at general practitioners or phase 1 units? For cancer screening, are you looking at specialty clinics, which could depend on the indication that you're after? For the treating cancer population, now you look at then at academic or tertiary care centers where they're following patients regularly. I think in general, the take-home message for me and what we have to think about operationally is, obviously does the site of clinical trial experience, which is first and foremost, but as importantly, do they have access to medical records whereby they can either access them for where they're being referred to or within their institution because the individual is being followed within their institution. And so data capture opportunities is very important in terms of when you're evaluating sites or the types of populations that you're trying to enter into your clinical trial.
Yeah, and that's definitely like something that we encounter a lot, even with or particularly with our, early stage detection studies. But I think, yeah, we'll definitely get onto that in a few moments. And are you guys maybe use as an opportunity to introduce a little bit what Harbinger does and your products do? Can you sort of tell the audience, where your products sit in this pathway, and then maybe where the focus is today, and then the longer term focus for Harbinger will be?
I'll jump in, Eric. You feel free to jump in as well. I think our test is a one that is based on the biology that understands many of the core elements of cancer, which includes metabolic growth, clonality, basement membrane invasion, and as a result, it is designed to understand the convergence of many signals that come into one for multiple cancers. So we are a multi-cancer platform, and that multi-cancer platform is biologically designed to focus initially on the epigenome, but is orthogonal to additional tests coming in. And so together, then we are able to really focus on multiple places in this continuum. And we've decided to lay our initial looks at where we can offer clinicians the best results in the asymptomatic average risk population, the high risk asymptomatic population, and then in the diagnostic population. I think that's where we are kind of focusing our first tray. And then, of course, not forgetting that there are many cancer patients who are already suffering and having to go through a process. We want to give them an opportunity to be able to be better diagnosed if we can. But for the moment, we're just looking at the upstream. And that epigenome is giving us a signal that allows us to kind of be able to offer tests in that space, which will be coming on the market, we hope, in the not too distant future. Eric, I don't know if you want to say anything more to that.
I don't have much to add except that what Hutan has outlined is it's allowed us an opportunity in the operations team to not have to focus on one indication. So we're going sort of a pan indication approach across multiple oncology indications. And so the only thing that I'll mention is that that has its pros and cons. The pros is that we can go to a wide range of sites. The cons is that we go to a wide range of sites. We're going to focus on one or two indications. And so, that's all I have to add there.
Nice. Cool. Well, yeah, thank you both. So yeah, maybe thinking about liquid biopsy studies in particular. Obviously not all. I mean, I don't think there's any two diagnostic tests in this space that are even barely the same. Tests have different analytes. They have different sampling procedures and so on. Maybe could you guys just briefly dive into the science behind these different approaches, and then maybe comment on how these different approaches kind of flow through into some of the operational decisions about sites, trial operational design. So yeah, I think Hutan, if maybe you could start with the science behind these different approaches.
Yeah, I mean, it'll be my absolute pleasure. So, of course, the North Star for any diagnostic is to have the least impact on tissue collection and the most impact on diagnostic yield. And so ultimately, the smallest amount of tissue, whatever it may be. And so we've moved for a paradigm of, and by the way, I'm trained as a surgeon, where we moved from a paradigm, the gold standard was big chunks of tissue, to actually where we can now move to much more nuanced sample types. And those other, some of the ones that you listed are blood, urine, and saliva. And of course, even gases like VOCs, volatile gases, organic gases, are also up there and are considered as being of utility. The nature of what we do is based on ctDNA, But of course, there are lots of orthogonal tests that we are also looking into and others are looking into, which is, as you say, EVs and proteins and RNA and multiple analytes. And this is where the multi-omic comes in. I'd say we found blood to be a particularly good source for what we need, which is DNA, because it allows us to look at methylation, it allows us to look at mutations, and it allows us to look at fragmentomics and proteins. So I think it is a very rich source for any company coming to or academic wanting to come into the space. But we are open to lots of the other sources. So, for example, I know that depending on the nature of the pathology you want to get from urine, which is typically a lot cheaper, very repeatable, and non-invasive, is that the type of DNA that one might get in terms of cell-free DNA is maybe less than 100 to about 150 base pairs long. It does give some methylation and some proteins and a few metabolites, but then a little bit harder to look at in the urine. Again, saliva is particularly good at maybe proximal viruses, maybe in the oropharynx and aerodigestive exposures. But again, it is not as representative of the whole system. And so when you're picking your approach, depending on what cancers or what diseases you want to focus on, your carrier or your sample type will be determined in that. I think the technology looking at blood is particularly advanced, whereas at urine and saliva is pretty hard. We've got much better at that in the past maybe 5 years, but to take that to a place where it's scaled and then being used in the clinic and so validated, and then gets to the kind of studies that we're looking at right now with you at Lindus is still in the experimental phase, whereas with blood, we're actually quite advanced in this space. So I think it is a useful platform for us to look at things, and that's where we are spending a lot of our time.
Yeah. No, nice. And actually, there's two questions that have come up that I'll try and thread in. So, Kamala mentions that urine-based liquid biopsy is an emerging area of interest, and asks if you could please kind of comment a bit more on that if possible. So yeah, I'll put that to you guys.
Yeah, I just say, just like in the oropharynx and aerodigestive conditions of saliva, urine is particularly good at looking at urothelial, prostate cancers, and maybe to some extent renal disease. That said, there are multiple metabolites in the urine that are reflective of systemic disease too. And so there's been a lot of studies where people have looked at actually VOCs, aromatic compounds in the urine. And I don't know if you know the background that people used to know that their domesticated dogs, their pets, were able to smell the different diseases based on the urine that the individuals had. And so that led to a lot of people understanding that urine is actually a pretty good source for this. That said, that work, which has been known for a long time historically, that dogs could pick up different diseases in their kind of owners, is a thing. But then to be able to do repeatably in a systematic fashion, people have looked at the organic aromatic compounds in urine, and it does give you an insight for some diseases, but it's not particularly ideal for others. Whereas blood is systematic, it goes to multiple organs. It is, it does get replaced quite easily, as does urine, by the way, but it does get replaced quite easily and does give you a feel in terms of richness, and both metabolically, but in terms of DNA and nucleotides, in such a strong way that actually it seems to be our richest source. So in the same way that we are currently using lots of fossil fuels and we're going to go into electricity, etc., I think we're still in that point where blood is very, very high yield for us, but we're hoping to move to the lesser invasive approaches as we move forward.
Nice. And there was an interesting question here. Again, I'll try and thread it in about I think about just based on your intro to Harbinger's approach, but anonymous attendee asks, how do you go about cost-effective pan-cancer validation, given the need to have statistical relevance for each single cancer type as well? So really about the, how MSED companies approach, validation given the basket of cancers.
Yeah, so I think everyone in the multi-cancer detection space is here for a reason, that when you have one test that can tell you the aggregate overall outcome for cancer, yes, no, but then at the same time give you a focus on multiple individual cancers, it does allow there to be an efficiency in decision making and an efficiency in terms of testing that was never there before. So that historically, if someone had some symptoms, let's say shortness of breath and a cough, they might go and get a bunch of tests that might be focused on lung cancer, and the fact that there might be an association with some of those symptoms and gastric cancer wouldn't be the first thing that people do, and so therefore they would go down the pathway of lung cancer, that would be excluded, and they have to come all the way back and then go down the pathway of another cancer, then maybe come all the way back, and maybe on the third go they'd find that they're relevant for GI investigations. Where the benefit of a multi-cancer is, hang on, in one go, you were able to dissect out that problem there and then, and as a result, that offers you efficiency both in terms of testing, but also in terms of downstream interventions, but also in terms of minimizing false positives, false negatives. That is the goal of multi-cancer detection approaches, either in screening or in diagnostics or in MRD. That's there to get that to happen is where we are all at, and focusing on making that something that is tangible for patients and doctors as we move forward.
Nice, thank you. Yeah, Eric, I don't know if you want to comment on some of the kind of downstream consequences of these approaches in terms of, just the operations of trial design. Yeah.
Yeah, and I know there's a couple more slides following this that talk about more of the operational considerations. I'll just say that, generally coming from the therapeutic space, these liquid biopsy protocols are typically much less complex. When we evaluated a lot of different EDC systems like Rave and IBM and Viva, we didn't think we needed all the bells and whistles associated with those very high, costly $500,000 systems. We can get into a little bit about what Lindus provides and Citrus, but that's not the point. The point is that these studies are much less complex. The biggest issue from my perspective is sample logistics. When you're dealing with typically fewer sites, but larger ends in the thousands, sample logistics is really important. And I can get into some of the things we've seen, which is a little bit alarming when you're dealing with thousands of samples with the types of errors you're seeing from sites. And so we can get into that in a few more slides, I think, Mary.
Yeah, we've got
the other take-home things is that the site budgets are much less than a therapeutic trial. The startup times are much quicker as well. And so just some things to unpack a bit more.
Yeah. I'll maybe just thread in one more question, then we have to move on. But yeah, Paul Billings asks to you, Hutan, yeah, are you talking about whole blood versus serum or plasma for your studies? As the latter, I think, can add complications. Yeah.
Yeah. I mean, we have a paradigm where we can look at whole blood and then we can spin it down to look at other things. But it really depends on what you're looking at. Some things are focused on hemoglobinopathy cancers, and therefore you can, focus on that. Some things are focused on things that are also in the serum at the same time. It really depends what your need is in terms of what analyte you want to look at or biomarker you want to look at. But either way, they both offer value.
Nice. Cool. So we'll go ahead and move on and make sure that we cover the other questions that have popped up in time. But yeah, so just to kind of set the scene, there are some three big challenges that we see commonly crop up and when designing and executing diagnostic studies. You've got accuracy, essentially balancing sensitivity and specificity. But the big challenge here is there aren't usually well accepted standards, and it often involves a bit of back and forth with the regulator. The second being clinical utility, and you guys have touched on this a bit already, and how it fits in the pathway and sampling procedures. And then finally, patient access, as we discussed, particularly at early parts in that pathway and the screening in kind of the healthy population screening part. We often see studies with very large volumes of patients to make the stats work given detection rates. Studies with 10, 20,000 patients, sample donors are not uncommon. So yeah, Eric, I would love your comment on some of these challenges. How do you think about balancing and solving some of these? How do you balance, accuracy, clinical utility? How does all of this add up to a study design that can be operationalized effectively?
Yeah, thanks, Mary. So as I had mentioned, I think typically some of the liquid biopsy trials will be less complex than an interventional study. It might be a one visit and done approach, or there could be one or two longitudinal samples, but the data collection is a bit more minimized. The safety database, the pharmacovigilance portion is fairly slimmed down or none. There are not individuals that are being seen for multiple weeks or months at a time on an interventional therapeutic investigational trial. When we set up our trial with Lindus, we wanted to focus firstly on sites that had clinical trial experience, extensive clinical trial experience, and diversity. It's very important from an access perspective. And we again wanted to make sure that they could find the different indications that we were targeting. We did put an emphasis on EMR access so that we could collect appropriate data and source verify that information. And also really wanted to focus extensively on sample logistics. Some of you who might be entering into the space might have temperature control issues, but we found in a study that we conducted previously a couple of years ago in about 10,000 subjects that samples were coming into Harbinger mislabeled, different dates of birth, samples could be hemolyzed, they didn't draw enough blood for us, the lab manual wasn't followed. And when you're talking about thousands of samples, even at a 5% error rate, it could get into hundreds of samples that you have issues with. And that was not appropriate for us. So we spent a lot of time at Harbinger really trying to make this process as simple and easy and understandable as possible for every site that we're working with. So a lot of effort has to be put into that dynamic of these types of clinical trials. And so we can get into some of those things that we did, Mary, if there's opportunity or if there's a question about that. But I think that's a really key thing to focus on is just sample logistics, because you are dealing with so many thousands of subjects and samples.
Yeah, it's a really good point, and you know, we see this a lot where yeah, any small mistake or ambiguity in like the sample handling protocol can get magnified so quickly. I mean, maybe if we had maybe just would love Eric to hear like what are like one or two tips you've had to kind of avoid those
Yeah.
scenarios and make sure the instructions are as clear as possible. Yeah.
Yeah. So at a previous trial, my team wasn't part of the study, but and then this is just a typical sort of easy error. You would think that your demographic information that's put into your EDC system will match the blood tube coming in to the organization or the lab that you're using. Well, inevitably, patient ABC in the EDC is ABF on the tube that you get internally. And so how do you make sure that that sample is the appropriate subject and is it tied to the EDC system? And the amount of time and energy it takes for just one sample to rectify that issue at the site level with your CRA, with your CRO, and with your lab is extensive for only one sample. Multiply that times hundreds of samples over a 10,000 subject trial, you get into a really large FTE effort. So we have used some technologies whereby when sites are entering information, we have a blood kit that they're able to sort of barcode scan into a system that talks to our EDC, and that information automatically transfers over to the Citrus platform within Lindus. And so the error rates have decreased extensively in this next study that we've performed, which has really been great. It doesn't take away from the fact that in many of these populations, you could have venous access issues, and so you don't get as much sample as you're hoping for or anticipated, and those are inevitable because those things will always take place. But it's the labeling issues that surprise me oftentimes because they happen more often than you would like or would think.
Yeah, yeah, makes sense. There was actually just one question because it pertains to the previous slide that I want to try and thread in. And so Roberta Carbone, apologies for the pronunciation, asks, in the analyte focus list, CTC, which I believe circulating tumor cells, are not mentioned. Is there a reason? Can you guys comment on CTCs? Yeah.
No, I think it absolutely should be there. We could be quite exhaustive in multiple kind of different cell types, antigen presenting cells, dendritic cells. We could put a whole tranche there. I think that was just a placeholder, but absolutely right, Roberta. And they very much figure in our mentality and many others too. Good call.
Nice. And Nora Al-Azam asks, how do you control for error rate, which I believe corresponds to just the sample handling procedure. I know you mentioned a good anecdote there, Eric, but yeah, anything else you'd flag? Any other advice for folks running these studies?
We've factored in an error rate of X percentage statistically, and so that's how we've sort of combated that. I will say though, I think if you're just very diligent on how you're training sites, making sure you're retraining sites that have issues early on, making sure that they're following the packaging instructions. It's just an ongoing, almost daily oversight in communicating with your lab personnel about how those samples are arriving and not a week or two later. We have a very fortunate opportunity that our lab is right here in our offices, so I converse with the lab people every day. If you're working with an outsourced vendor, you have to spend a lot of time on making sure the logistics there are appropriately understood and the communication channels.
Cool. No, thanks so much. We should probably keep it moving. But yeah, we've touched on this already, so maybe no need to dwell on this too much. But you know, everyone kind of defaults to a sort of drug pharma paradigm for clinical trials. As we mentioned, diagnostic trials, very different, and within that, kind of liquid biopsy trials have their own unique flavor. But yeah, Eric, maybe if you could just comment here on some of the other big differences, perhaps some of the common mistakes that result from misapplying a pharma drug trial paradigm, in the diagnostic space. Yeah.
Yeah, so again, I think what you're dealing with here is most likely a lower number of sites, but a much higher volume of subjects or patients. And so what does that mean? Well, do you really want a site to enroll 100 subjects in 2 weeks? Can they actually provide quality data? So while you want high enrolling sites, making sure that they have the capacity to be able to provide that quality, clean data. So that's one consideration. There's much faster startup timelines in this environment than it is in a therapeutic study when you're working with academia. We are working with a couple of academic sites, and we've been up and running for a few months, but those academic sites still aren't up and running. And so you don't have to necessarily, unless your executive team wants you to or needs you to, have to go to those KOLs that are sometimes very prominent in oncology therapeutic studies. The price point's much lower per subject, especially if it's just a 1 or 2 visit study. And one thing I've always focused on as an operational person, while I'm not an MD, I'm always trying to challenge the team on capturing what is going to be analyzed in your clinical protocol versus, so what's must have versus nice to have. And so we've had some good conversations about that internally, and I think simplifying the protocol as best you can while capturing what you need is very important. And so those are just some key takeaways from me, having come from the therapeutic space and into this liquid biopsy space.
Nice. And actually, there's a really cool question that really hits home for me from Adam Benson, who asks, yeah, small biotechs almost never have the trial budget of pharma. So how do you balance these challenges while also needing to maybe bootstrap your program, especially given the fundraising environment? And in particular, how do you deal with sites often having a kind of pharma level compensation expectation, but then only being able to pay them, less, or just, diagnostics procedures are often way less complicated, right? So how do you how do you manage expectations of sites?
I'm chuckling here, Adam. Thanks for the question,
Yeah.
because it's been my life probably for the past many years. At Harbinger, we did have a budget in mind, and and I think again, there's a future side to talk about this, but we're a small organization with less than 100 employees. We evaluated a lot of global partners for what would be our entire platform. Name the top 5 CROs. We evaluated all of them. We also evaluated Lindus. It wasn't necessarily that the price point was significantly different between the CROs. The reason why we partnered with Lindus was a bit more fundamental from my perspective. One was that Their outsourcing model, and Mary, I don't want to speak for you. I'm just giving you my perspective.
Go for it. Better,
The
yeah,
way
better
the way in
for
which they
you.
allocate their teams is much more on an FTE basis instead of 0.2 of a PM across 17 different roles. We have a very small, tight-knit team that are extension of my team here at Harbinger. They're available to me on a everyday basis. It's not that they're focusing on 6 different trials for a percentage of their time. I really like that model. We also have a milestone-based payment with Lindus. They were and Mary, I'm sorry to say this, they were in our negotiation. They were very reasonable and were eager to show us that they could perform. So we entered into a bonus penalty clause with Lindus. Now that might not be something that your organization wants to do, and it might not be the right environment for Lindus either. But I don't think it hurts asking those questions. And I think what that fundamentally shows to me, and I think the executive team here, is that you're working with a partner who is truly your partner, who's in it to win it on the execution. Not to coin a phrase, but that's what we felt. We felt like another fish in a big pond with some of the larger CROs. And again, that's just for us at Harbinger. It might not be your unique situation, but for us at Harbinger, that's how we felt. And so I'm not sure if I answered your question, but so one thing I didn't outline and maybe answer was the costs and the negotiation process with sites. We were very comfortable pushing away from the table, as they say, if sites came to us that were 2, 3-fold of their competitors. And I think you have to be willing to do that, as long as you're choosing sites, again, that still have clinical trial experience, has the patient population, and have the staff that can support quality data. And so, but you have to be willing to walk away and actually potentially negotiate with your executives internally about walking away from some particular sites that they might think you should be working with. And so I've always tried to represent operations from an execution perspective, making sure that fundamentally you can execute on the trial with clean data and quality sites. And so hopefully I've answered that question.
Yeah. No, thanks. And thanks for the kind words. And maybe I just add from our perspective across, our trial portfolio is, you know, there are kind of readily available benchmarks for different trial procedures that you're asking the sites to perform. Diagnostic studies, particularly in early screening, have the advantage of, these are not complex procedures, they're very standardized. And so we found it very useful to be able to quote, these benchmark costs back to sites and show that what we're proposing to pay them is, within the 50th or 75th percentile, i.e., upper tier of the cost with the appropriate, headroom built in. And from there, there's not most sites can't really argue back to that. But yeah, there's always the yeah, there's always the occasional yeah, kind of high level site that has their own ideas. But yeah.
I will say one more thing, Mary, that we as an organization made it determined to under promise and over deliver. And so we made the decision to include more sites than we thought was actually needed to meet the enrollment timeline, because the startup costs were so low, sometimes less than $20,000. And we added about 7 more sites than we thought that we needed. Well, what's actually happened in the first few months of enrollment, we are about 75% ahead of our enrollment projections, which has now caused some issues fundamentally with other functions within our organization. But there's no better feeling as an operation leader to show your internal team and for Lindus as well, that we are far exceeding our enrollment expectations. And for any organization bringing in a potentially pivotal trial 3-4 months before you had anticipated is worth that $10,000-$20,000 startup costs at those 7 additional sites. And so that's the mindset that we had going in, and I think it's worked quite well for us.
Nice. Yeah, we're obviously super stoked about the enrollment progress as well. Yeah, it's great. Cool. So yeah, maybe moving on to considerations if we're thinking specifically about MSEDs now. So in early screening, yeah, there are often statistical challenges, where you're setting your kind of target on the sensitivity specificity curve, and of course, operational complexity. Yeah, maybe just Hootan, a general question for you, and we have some specific questions from the audience on this. on the the statistical side, like how did you, in as much detail as you can share, how did you kind of think about the statistical objectives for your study, and how does that relate to your product's intended use and the type of cancers it detects? Yeah.
Well, it's a great question, and I guess firstly, just comparing ourselves to the statistics and powering uh, of a therapeutic trial. I mean, there is a fundamental difference in philosophy, and the philosophy for therapeutics is essentially, does a intervention work in individuals, or this number of individuals, to the certain extent. Whereas for the diagnostics, above and beyond just the diagnostic accuracy as our outcomes, which is very clear, it's ultimately whether does our platform or our product help doctors make better decisions that then in turn improve outcomes. So there's that double hierarchy that is always at the back of our mind as we define our primary and our secondary endpoints. So that's not to say that we don't always have diagnostic accuracy, which we typically do, but in and above that is what is the utility of that diagnostic accuracy. And so that is one thing to think about. The other thing is, of course, we are in a multi-cancer detection space. And increasingly, when we go to regulators, the whole point of a good test is to identify an individual suffering from a condition, but at the same time to minimize what is seen to be quite a heavy lift with a lot of diagnostics, which is they have very high real-world false positives or false negatives. And as a result, those two elements, that falsity is becoming a heavy lift for a lot of products that can kind of pick up a lot of things, but also in proxy have lots of problems in terms of picking up unnecessary things or missing actual disease. And so what we have already talked about in as a company that we want to kind of spread out to the wider ecosystem is not just to have diagnostic accuracy as our endpoint. And remember, when people present diagnostic accuracy, they look at aggregate diagnostic accuracy. So for many multi-cancers, they go aggregate sensitivity and aggregate specificity and aggregate PPV, et cetera, and NPV. The nature of that is fine, but then someone will say, you might be weighting too heavily in terms of one cancer or another. And so many of the metrics already out there are able to tell you if a test is good at picking someone with cancer, yes, no, we can pick a cancer, this is the case. But they haven't yet been able to produce something that we call intrinsic accuracy and offer something called tissue-specific performance capability, which is how good is that test on aggregate per cancer? So that if I have a cancer test that is designed to look at brain, thyroid, and prostate, how good is it? And I'm just making that up on the spot, by the way. How good is that test as overall accuracy? And how good is it per cancer test without me having to list out the accuracy for each of those cancers, because it's very difficult then to compare and contrast across 55 or whatever, 100 or 25 cancers, etc. So there is a statistical way of doing that. We've kind of talked about it, as I said, called intrinsic accuracy, which is able to weight across multiple cancers the accuracy in terms of specificity, sensitivity, PPV, NPV, etc., as a single number, how a particular device or platform can work in multiple cancer spaces. And then we use that TSPP approach to really identify how many individuals we have to have, what is the minimum number of individuals we have to then achieve our evidence goals. And this is exactly what we did when we went to Lindus, our friends at Lindus, to be able to say, these are the number of sites we need, these are the potential sites of, numbers of individuals we needed at those sites. I think that's probably our biggest element in terms of study sizing here.
Nice. Having said that, actually, there was a very specific question from Harold Garner, and which I want to try and thread in. So he says, at AACR, Harbinger Health stated, actually, I'm not sure, essentially, he's quoting your statement at the conference, when MLX and, well, actually, 8, my Roman numerals are correct,
Yeah.
were both set to targets. Can you see the question, or?
It's from Harold. Hello, Harold. Yes. So,
okay, cool. Yeah.
The work done here was based on something called inter-individual subtraction, the IIX, which is a process to be able to, if you have a signal noise ratio from cell-free DNA in the bloodstream, That comes from cancers, but can come from other sources, including polymorphonucleocytes in white blood cells. And we have formulated a way to remove the signal from the white blood cell cfDNA, and then to be able to, as a result, have almost an enriched way of looking at cfDNA with higher PPV and high accuracy. And that's one of our innovative moves forward that we're hoping to get into our products in due course.
Thank you. No, that's extremely cool. I think we had one other kind of relevant question on the stat side. So yeah, Alia Parvez asks, how do liquid biopsy trials account for tumor heterogeneity and the possibility of false negatives, particularly in early stage cancers?
So that's an excellent question. By definition, cancers themselves are heterogeneous, and multiple cancer companies are not only working hierarchically on each individual cancer being heterogeneous, but the stages that they present being heterogeneous, and then on top of that, having multiple cancers that are also in the mix. And so I think the way we minimize that is having our assay to be particularly focused on big chunks of the epigenome that really allow us to be understanding the common cancer message, but also our novel AI approach to be able to tune to that. And that is all about before working with our friends at Lindus in a study that we're doing at the moment. We've spent many years doing studies that aren't trials, looking at cancer signals in individuals, lots and lots of different cancers, lots of different populations, and lots of different settings in the United States, but hopefully lots of different ethnicities, lots of different socioeconomic classes, and ages and BMIs and demographics, to ensure that our test is equitable and that is minimizing bias in any particular subpopulation. And this has been one of our biggest intents. So we have a test that is a sequential test that allows us to kind of have a high MPV rule out and then a high PPV rule in. And all of those tests are designed to be in a wide range of population, a wide range of ethnicities, etc. And the way to mitigate that, there's no easy way of doing it other than actually getting good data across those populations, which is what we've done. I hope that helps.
Nice, thank you. Cool. We are rapidly approaching end, so I'm going to try and canter through the remaining slides so we can answer as many of the audience questions as possible. I think we've touched on actually most of the topics here. There's maybe one or two that I wanted to call out, which was yeah, the comparator and EMR hurdles. And we have a couple of interesting questions on these as well from the audience. So maybe can one of you just comment on the comparator selection, because this is something we see coming up with some of our sponsors. Yeah.
Yes, I'll jump in with comparator and Eric come in as well on the ops. In terms of comparators, in diagnostics is always interesting because the classic diagnostics paradigm is to compare your test to what is called a gold standard. So you have an index test, and then you have a gold standard. You're meant to compare the two, and then you come with your matrix of doom to appraise that. In reality, there is a wide range of comparators. There is a composite comparator of multiple different standards being used. In some very specific cases, you might be able to compare yourself or oneself to, let's say, a PET CT or a CT scan, etc. The reality is there's a lot of need for Bayes and instruments in 2025 and beyond, because we are sometimes having a test that is more statistically capable of picking up tests at different thresholds than what the gold standard already exists. And so therefore we have to have kind of a statistical methodology using Bayesian approaches to really identify non-inferiority, superiority in a way that we can find an evidence base that can really highlight the strength of a particular assay of one type or another in this multi-cancer space. So I think that ideally you want a straightforward comparator that makes the maths and the presentation easy, but in reality, we have to use the comparators of a clinical pathway of care rather than a single test typically these days. I hope that helps.
Nice. And then, yeah, there was another point I wanted to pick up on here on the EMR. So, yeah, this is a really common challenge in oncology diagnostic trials. You know, patients are sampled at a given site. Critical that you get their downstream diagnosis and as much data from them as possible. but that diagnosis might happen at a separate site with a separate EHR system, et cetera. Eric, can you comment on that? And then there was a cool question here from Sarah Wise that I want to thread in as well, but I'll let you comment first.
Yeah, it's a very real issue. A typical example is if you're doing a screening trial, perhaps, and someone who's undergoing a colonoscopy, they're getting the colonoscopy done at the GI specialty clinic. Perhaps there's a suspicion they go away, they get diagnosed with colon cancer in 4-8 weeks. Don't know where they're going at that moment. Can the site actually retrieve the diagnostic information, the staging information, any other pertinent diagnostic information? And I think it just comes down to site feasibility and really honing in on where typically their patients are referred to, what type of relationships they have with those referral sites in their network, perhaps. Sometimes you can find sites that they're intertwined, so it's more of a specialty clinic that also deals with a therapeutic environment, so that the patient can go down the hallway and be treated for the colon cancer. And so it's just really making sure your site feasibility is really robust. And in the site feasibility questionnaire, making sure you're focusing on those set of questions that really make you feel comfortable that you're going to be able to retrieve that information. Because at the end of the day, if those data points aren't retrievable, what does that mean for your quality of data? It's pretty impactful. So making sure the CRO you're working with has a really robust feasibility questionnaire and program, and that you're involved heavily in that is extremely important.
Nice. Yeah, and this kind of leads us nicely to Sarah Wise's question, which is, Eric, you mentioned emphasis on EMR access. Does this mean that you only worked with sites who would provide Harbinger or Lindus direct access to EMR to collect data, or or work with sites that that had to enter data at site? So yeah.
Yeah, we did focus on making sure we had EMR access. We're doing a lot of remote monitoring, which also helps. And so we did try to make sure that all of our sites had really robust EMR access programs. We're actually uploading a lot of pathology reports and even images if required in our EDC. And so we can have that source documentation, obviously de-identified, but we also spent a lot of time making sure we had that capability as well.
Yeah, I just add, Sarah, that remote EMR access from us as the CRO's perspective, a lot faster, a lot less expensive to monitor, because we're not flying monitors out the site. Doesn't mean that we don't do site initiation visits, interim monitoring visits. We still do those, but those can be more focused on value add things like building relationships with investigators, checking, to Eric, your earlier point, that sample handling protocols are being followed, those kind of things that we can only do in person. So yeah, it's a remote EMR access is a huge win for us in these studies. Yeah.
One other thing I didn't get into, Mary, and we fortunately don't have to deal with it here at Harbinger, but maybe some other organizations do, is temperature control. When you're dealing with thousands of samples, if you do have a product or a sample that needs to be temperature controlled, that obviously creates a whole another logistical consideration. Hot climates, cold weather. In our previous study, we had higher incidence of hemolyzed blood samples showing up at our lab in really cold months. And so making sure you're temperature controlling that environment, it can cost a lot of money, but it's also important consideration, or temp tail monitors or specialty couriers and so forth. And if you're going globally, that's a whole nother set of circumstances and logistical issues you have to keep in mind.
Yeah. And there's actually, what I might do is, because we're nearly out of time, I do want to touch on some of these remaining points and a couple of the questions. We can just launch the poll in the background and just have that run. And in the meantime, I want to come to, there's a very interesting question here from Amy Isaacson about decentralized models. So curious to hear Mary and Eric's take on the use of a decentralized model and mobile phlebotomy in large liquid biopsy studies. I can comment on this with Eric. I don't know if you have any comments first, or yeah.
You can go first, Mary. I can chime in a little bit.
Okay, cool. Yeah, I think this is super interesting because this is something we have a lot of experience with. I would say, big learnings here are this can be a very powerful tool in your arsenal to greatly reduce cost and reduce timelines for these large scale liquid biopsy studies. However, the decentralized model needs to be deeply integrated with the rest of trial operations. For example, I mean, Eric, you were just talking about sample handling. We wouldn't really propose, or we wouldn't we would be loath to consider a decentralized model for a sampling procedure that required immediate spin down and had, low tolerance on, required temperature control, shipping or frozen shipping samples. Because, I mean, particularly if you're sampling patients at home, there's a high risk that something could go wrong if you have a large variety of nurses, traveling to patients' homes, carrying out samples. Where it has been a huge win for us and for some of our trials is, large studies where, there's kind of a very simple sample handling protocol. We're, enrolling large volumes of patients who may be, disparately located, or where we use it as an adjunct to site-based visits. So we might have, an initial visit at a site where, more blood is drawn and more data is taken from the EMR, and then maybe there are follow-up samples that we can easily do from home. So I think, it's definitely not one size fits all, but where it can be used, it can have a massive impact on costs and timelines. And so we always look at it on a case by case basis, but it is something that we have, we have full decentralized capabilities in-house. So yeah, I hope that helps. And I don't know, Eric, if you have anything.
Yeah, no, I think I couldn't have said it better, Mary. We haven't really explored this model here at Harbinger because we felt as though we could enroll the subjects within the sites that we chose. However, I agree with underserved populations. I do think that's an opportunity. I will say that mobile phlebotomy, the price point's going to go up for you. But it is a nice way to potentially, as you had mentioned, Mary, for follow-up visits or follow-up access of information. That could be a nice approach.
Hutan, I think you have your hand raised.
Just briefly on that also, I've also worked previously on remote phlebotomy with some devices that have worked well with both our CRO friends and have been able to achieve in a vaccine situation, many thousands of individuals. That again, this is my role at Flagship. That one of our portfolio companies had a device that goes on your take the capillary blood without a blade, and so I that was very, very useful, and I think maybe the way forward in many instances.
Nice. Cool. And we are nearly done. So I'm going to share, the results of the poll. I'm also then just going to very briefly hand over for key takeaways. And we still got a lot of unanswered questions. We will do our best to answer your questions async. So thank you very much. And really appreciate everyone's questions. But yeah, now that we're coming to the very end, Hutan, Eric, quick fire. Maybe if you could each just highlight your key takeaway from the discussion today. Yeah.
Go ahead, Hutan.
Yeah, Eric, go first. Go for it.
Okay.
Hey, listen, first and foremost, thank you for the opportunity. I'm available on LinkedIn if someone has some offshoot questions. But for me, I think it comes down to choosing the right sites, great feasibility, picking your right CRO partner for the right price point. I know there's a whole bunch of things that always should be considered, but sample logistics is a huge criteria here. Make sure you're spending time on sample logistics. You can't just have samples showing up without a plan in place for rectifying issues. And so that's a really key takeaway.
Nice.
Diagnostics is about understanding how decisions are made to make changes in patient outcomes. It's not all just purely about diagnostic accuracy. Think about the clinical utility. And when you pick your CRO and pick your team to run your evidence base, always at the back of your heart, make sure you've got that clinical utility in mind.
Awesome. Cool. With that, we are out of time. So thank you all so much for listening, for contributing questions, and above all, a huge thank you to Hutan and Eric of Harbinger Health. I'm very excited to have you with us. And we will send over the slides remotely. We will also do our best to answer all of your remaining questions. So yeah, thank you again and have a great afternoon.
Many thanks to Lindus. Many thanks to friends of Lindus. All of you. Bye-bye. Take care.
Thanks everyone. Bye-bye.